Students, welcome back. Uh, in the previous topic, we have completed the fertilization, the process of fertilization we have completed. And you know very well, in the sexual reproduction, fertilization is having a key role where we have completed the development of a zygote where two haploid cells, the male gamete and the female gamete fuses together to produce a diploid structure that is called as zygote. And in sexual reproduction, the fertilization is a key event which is going to differentiate the sexual reproduction in three steps: the pre-fertilization, fertilization and the post-fertilization. And here, after completion of fertilization, we are beginning with the post-fertilization event. In case of humans, all the post-fertilization event can be considered or can be studied under a single head that is called as the embryonic development. Are you getting my point? All the post-fertilization event in case of humans are considered or can be studied under a single head that is called as embryonic development. In embryonic development, the zygote or the activated egg is going to undergo in the process of the embryonic development, where so many processes are there like the cleavage, morulation, blastulation, gastrulation, organogenesis. So these four processes are the basic processes where the zygote is going to convert in form of a multicellular structure or embryo. So we are just going to start with the first process that is called as the cleavage. What happens? The first process just after the formation of the zygote undergoes in the process of the cleavage. So just start the cleavage. So we are starting with the cleavage today. So what is cleavage? Cleavage is a series of repeated mitotic division. My dear child, just remember, the meiotic division is limited only to the formation of the gametes only. The meiotic division will present only during the formation of the gametes, whether it is male gamete or female gamete. You have observed it well during the formation of primary spermatocyte to secondary spermatocyte and secondary spermatocyte to the spermatid. The meiotic division was there, and similarly there in case of oogenesis, secondary oocyte and the egg cell are being produced through the meiotic division. But rest of the time all the cells of the human body or the animal body are undergoing in the process of mitotic division, because here we need to conserve the diploid nature of the cell that we have received or that we have restored during the fertilization event. So here during the just after the formation of zygote, the zygote will enter directly in a series of continuous repeated mitotic division. Again and again the mitotic division will be there. So the zygote will enter in a series of continuous mitotic division that will lead to form a multicellular structure from a unicellular zygote. Zygote was made of a single cell because it was fusion of a male and female gamete. So zygote was a single cell structure and it has undergone in the repeated mitotic division, in the series of repeated mitotic division, and will undergo in the will change in form of a multicellular structure. One very important thing, even though zygote has undergone in the repeated series of mitotic division, then too the resulting cell is not going to change its size. The size of the zygote will remain constant. Size of the zygote will not increase. Size of zygote will not enlarge. The size of zygote will be constant, size of zygote will be similar, but the number of cell inside the zygote will increase. So here diploid to diploid cell will be produced from the activated egg or the zygote. So is it clear what is cleavage? Cleavage means simply the cleavage means cleaving. Cleaving means cutting. So cleavage is a process of cutting the zygote. So how the zygote is being cleaved down or how the zygote is being cut down? The process of mitotic division is cleaving the zygote into the two parts, then next cleavage, like that. So many of the cells are being produced after the formation of the zygote. The cleavage term was given by the Von Baer. This is important question for the NEET examination. The first time the term was used by the Von Baer. So here a series of repeated mitotic division in a fertilized egg, that is zygote, to produce the multicellular structure without changing its size is called as cleavage. Remember, without changing its size, just now I told you there is no change in the size of zygote, only number of cells are going to increase. And the specific point, remember here, the cells which are produced after the zygote are called as blastomere. Whatever the cells are produced after the zygote are called as blastomere. My dear child, blast means the cells which are going to synthesize something new. The blast cells are the cells which are going to synthesize the new structure. They all are the totipotent cells, though it can change in form of any of the structure. So the resulting cells of the cleavage are called as blastomere, and these cells are taking the responsibility to develop the complete embryo. So is it clear now? Here the new cells form, that is called as blastomere, and the embryo is and the zygote divided again and again by the help of mitotic division. We are just going to discuss some of the properties of the cleavage. What are those properties? Just think. First property, the division of the division during the cleavage, that is mitotic division. Remember, again and again I am saying the division during the division during the cleavage is a mitotic division, and that mitotic division is rapid and quick. We are saying rapid and quick, and we are comparing, or we are going to compare that division with the help of normal mitotic division. So as compared to the normal mitotic division, the cleavage or the mitotic division occurs during the cleavage is rapid and quick. Very faster division is there. If the question asks you why this is so, the next property is there, because the interface in the cleavage is very short. Are you getting my point? The cleavage division or the cleavage is very fast and rapid process, very quick process. If question is there, even though it is also following the mitotic division, so then why it is quick or faster? Because the interface in the cleavage is very much short process. Interface is short. So once again the question will be there, then how the interface is short? Because the interface in the cleavage is having the involvement of only S phase. Only S phase is present, no other phase is present during the cleavage only. So is it clear? Division is rapid and quick. Why this is so? Because the interphase is short. And why interphase is short? Because the cleavage follows only the S phase. Means what? Only DNA duplication is there. There is no growth of the cell occurs, means there is no storage of material occur, like RNA and protein synthesis will not be there. Only the duplication of DNA is there. That is why what I am saying, the DNA synthesis is faster than the normal mitotic division. So DNA synthesis is faster than the normal mitotic division. No G1 G2 phase is present. G1 G2 you knows very well. The three stages are there of the interphase, G1 G2 and S. So no G1 G2, the growth phase is absent. That is why the cell size is not going to increase. So because G1 G2 is not present, G1 G2 is absent, so no change in the size of cells as the growth phase is absent. High consumption of the oxygen. Is it clear? So all these points are the properties of the cleavage. One very important point that I am just going to discuss you. Remember my dear child, whenever the higher questions are there, higher order questions are there, so remember the rapid rate of division during the cleavage is maintained by some of the proteins. Are you getting my point? The rapid rate of division during the cleavage is maintained by some of the protein. Those proteins are called as cyclins and cyclin dependent kinases. This is very important question for the NEET examination. The rapid division during the cleavage is regulated by some of the proteins. That proteins are called as cyclins and cyclin dependent kinases. So properties are clear to you all. Cleavage means what? Repeated series of mitotic division where number of cells will increase but size will not increase. The cells which are resulting from that one, that is called as blastomere, and by the process of cleavage a unicellular zygote will convert in form of a multicellular embryo. Properties: division is very rapid and quick, interface short, DNA synthesis is faster, G1 G2 absent, size of the cell will not increase, and rate of oxygen consumption is very much higher. So moving on to the next point. Here we are discussing the laws of cleavage. Laws of cleavage means to say some of the scientists, some of the researchers has tries to explain how the cleavage occurs. They are just going, they have tried to explain the basic principle, basic mechanism of the cleavage. So here some important principles we are discussing. First one is called as the Sachs law. Sachs law was proposed by, or Sachs law was presented by the Sachs in 1877. It was presented by Sachs in 1877. The Sachs has presented two concepts regarding the cleavage. The concept number 1, cell has to divide in equal daughter cell, means what? The cell has to divide into two equal cells, neither larger cell nor the smaller cell. Even though we will see again, we'll see ahead, this concept is not accurate, but at that time it was accurate. He has given the concept, the cell will be divided into two equal cell, two equal daughter cell. And remember, in normal mitotic division the cells are called as daughter cell, but in case of cleavage they are called as blastomeres. So new division plane intersect the previous plane, that is important point. The new division plane during the cleavage intersects the previous plane at the right angle. If the division plane is, first division plane is the longitudinal, then the second division plane will be horizontal from first one. So that will be a kind of cross structure, it will be a kind of plus shaped structure. One division plane is the longitudinal, then second one will be right angle to that, that means that will be an horizontal plane. So these 2 points were given by the Sachs. Later on the Hartwig has modified some points or some of the concept of the von Sachs, that is Hartwig's law. It was presented in 1881. He has modified the Sachs law. Just now I just told you, okay, Hartwig has modified the Sachs law only. He has modified, in microlacethal egg and isolacethal egg the mitotic spindle runs centrally. Microlacethal egg means what? When the amount of yolk is very much less, and isolacethal when it is equally distributed in the egg, the mitotic spindle runs centrally. So mitotic spindle means to say the spindle fibers which are driving the chromosome on the opposite pole. So these spindle fibers will run on the central position, it will run from the central region only. But in the telolacethal egg means when the yolk is concentrated there in the vegetal pole. One pole is there, that is called as the vegetal pole. So when the yolk is concentrated in the vegetal pole, then it is called as telolacethal egg. So when telolacethal egg is there, so that time it is closer to the animal pole. Why this is so, we'll see in the next law only. So Hartwig, what it says, Hartwig law, it says that in microlacethal and isolacethal egg the mitotic spindle runs centrally, but in case of telolacethal egg it is closer to the animal pole. Moving on to the next law, that is called as the Balfour's law. Balfour's law was presented by the Balfours in 1885. Very straightforward law. The rate of cleavage is inversely proportional to the amount of yolk. Rate of cleavage is inversely proportional to the amount of yolk. So what is the meaning of rate of cleavage is inversely proportional to amount of yolk? It means when in the egg we cannot say, in the fertilized egg we cannot say the yolk is present equally or throughout there in the complete cytoplasmic region. So Balfour's has stated that the cleavage is going to provide a negative impact over the rate of division. The yolk is going to provide a negative impact over the process of cleavage. Are you getting my point? The yolk is providing a negative impact over the cleavage. Negative impact means to say the region where the yolk is present will not undergo in the process of cleavage, means yolk is providing a resistance on the process of cleavage. So only part of the egg which is not having yolk, that will undergo in the process of cleavage. Suppose the centrolacethal egg is there, so the cleavage will occur on the peripheral region only. If the telolacethal egg is there, so the cleavage will occur on the animal pole only, not on the vegetal pole only. So are you getting my point? So if megalacethal egg is there, so only few part where the cytoplasm is located, there only the cleavage will be there. So this is very important law, and you can understand just above this one, the Hartwig law, what in the telolacethal egg it is closer to the animal pole. This is what the application of this one. Balfour's law states that the yolk is going to present a resistance on the process of cleavage. Where the yolk is present, the cleavage process will be affected. Next one and the last one that we have to discuss today, that is the Flieger's law. Flieger's law, again the modification of the Balfour's law. The mitotic spindle always elongates in the direction of the least resistance. You can understand well. Mitotic spindle means what? The division. If the mitotic division is going on, means the mitotic spindle will always run in the direction of the least resistance. Means what? It will run in the direction where no yolk is present, where yolk concentration is very less. Either it is absent or it is very much less. Means what? It will run in the direction of animal pole, not on the direction of the vegetal pole. So this is clear to you. Sachs, Hartwig, Balfour's and the Flugger's law. The important concept or important principle is of the Balfour's law, where the yoke is providing a resistance on the process of cleavage, or yoke is providing a negative impact on the process of cleavage. Now moving on to the types of cleavage. We can divide, or we can classify the cleavage on two basic types. First one is a fate of the blastomere and amount of the yolk only. So on the basis of fate of blastomere, you knows very well, the resulting cell of the cleavage are called as blastomere. So how the blastomeres are going to be used in the future, that are going to make, or that is going to make the basis of the classification. So first one is called as the determinate. On the basis of fate of blastomere, called as determinate cleavage. Determinate cleavage means to say when the blastomeres have defined fate in the future organism. Means what? If a blastomere is going to degenerate or going to damage, so the particular structure which is confined to that blastomere will not going to develop in the future organism. Are you getting my point? Suppose a blastomere is going to develop, or going to originate, or going to develop in form of the eyes, and that is going to degenerate. So the organism will degenerate organism will going to develop completely, but the eyes will not develop. So that is what the blastomeres are. The fate of blastomere is fixed and determined. Are you getting my point? The blastomere fate is determined. That is why that is called as what? Determinate type of the cleavage. Determined means to say each and every blastomere has their fixed fate. If the blastomere is not available there, so that structure will not develop in the adult form. So this is the property of proterostomia. Proterostomia means the group of animals where the blastopore, will see the blastopore is going to produce the mouth first. The example of that, nematoda, mollusca and annelida. Just opposite to the determinate cleavage, that is indeterminate. Indeterminate means what? Where there is no fixed fate of blastomere is reported. Blastomere का कोई future fix नहीं है. Each and every blastomere are considered as the totipotent cell. Are you getting my point? Each and every blastomeres are working as a totipotent cell, and they are having the complete capability to produce the complete structure of the future embryo or future organism. So that is why if the blastomeres are getting damaged, degenerated, break down, then there will be no effect on the further development of the embryo. And my dear child, this is only going to make the case of twinning also. Suppose during the embryonic development, if the blastomeres are getting cleaved there, blastomeres are getting separated there, so how many are there? How much number of the blastomeres are separated? Each and every one will get to produce that much new organism. That is what the case of twinning. Embryo फट जाता है in cases में, तो complete embryo is going to generate the complete blastomere is going to generate the complete embryo or complete adult structure. That is what the fate is not determined, and this is the property of deuterostomia, where the mouth instead of mouth, the anus is developed from the blastopore. And humans, we are following the indeterminate type of the cleavage. So humans are having indeterminate, means we are performing the totipotent nature of the blastomeres, and that is why we have the cases of the twinning. So moving on to the next type of the cleavage. Next one is on the basis of yolk. First one is based on the fate of the cells are blastomeres. Now the basis of the yolk, how much yolk is present, and yolk is how the yolk is going to influence the process of cleavage. So first one is called as complete holoblastic cleavage, complete or holoblastic cleavage, and second one is called as incomplete or the meroblastic cleavage. Complete or holoblastic cleavage means what? When the complete egg is going to divide, when the complete egg is undergone in the process of meiotic division, then it is called as holoblastic. Holo means complete, blastic means blastomere, complete, blastic, complete means complete egg is undergoing in the process of cleavage. So this is what, when the complete egg is undergoing in the process of division, then it is called as complete holoblastic. So complete holoblastic will be present in those eggs where less amount of yolk is present. Since you knows very well, as per the Balfour's law, yolk is presenting the negative impact over the cleavage. So here two types of the complete holoblastic cleavage is there. One is called as equal holoblastic, another one is called as unequal holoblastic. Equal and unequal holoblastic is based on the type of the blastomeres produced after the cleavage. When all the blastomeres are equal in size, then it is called as equal holoblastic. When all the blastomeres are equal in size, same in the size, then it is called as what? Equal holoblastic. characteristic of isolecythal egg, metatheria and eutheria. The mammals are there, metatheria and eutheria. Metatheria paused mammals, eutheria the normal true mammals. Unequal holoblastic, just opposite to that, when the blastomeres produced after the cleavage are of two different size. Some of them are larger, some of them are the smaller. So the larger one is called as the megamere or macromere. The larger one is megamere or the macromere. A smaller one is always called as micromere. So when the blastomeres are unequal in size, whenever the blastomere produced by the cleavage is unequal in size, they are called as unequal holoblastic. Megamere means the larger one, the blastomere which is larger in size, and micromere the blastomere which is smaller in size. This is present in the mesolecithal egg, and important that is present also in the humans. We are following the complete holoblastic but unequal. Okay, so two types of cleavage we have reported. In our case, in human case, what is happening? Indeterminate, complete holoblastic but unequal type of the cleavage. Second one, incomplete or meroblastic cleavage. Incomplete or meroblastic cleavage means what? When the yolk is present in the most of the part of the egg, means what? When the yolk are when the eggs are heavy yolky, large yolky eggs are there, so the yolk will present a negative impact. So in the region of yolk, the cleavage will not be there. So this is what the megalecithal egg. So only the non-yolk part of the egg will divide or will undergo in the process of cleavage. So it is also of the two types. One is called as discoidal meroblastic and one is called as superficial meroblastic. Discoidal meroblastic is the property of the birds, reptiles and the prototherians. Discoidal meroblastic is the property of birds, reptiles and the prototherians. What is discoidal? The discoidal meroblastic is present there in the cleavage where we can say discoidal means what? A disc like portion of the yolk is present, and above that portion only the cleavage will be there. So restricted to a disc of cell cytoplasm in the animal pole. Superficial meroblastic. Superficial meroblastic means what? The centrolecithal egg means what? The complete yolk is lying in the center only. So whatever the cleavage is there, so that will be a peripheral one. बाहर की तरफ cleavage होगा, अंदर की तरफ cleavage नहीं होगा. So that is what called as superficial cleavage or superficial meroblastic. So is it clear to you? Now moving on to the human cleavage. Even though we have just seen what is the human cleavage, what type of the human cleavage is there, so but to understand few more points about the human cleavage, remember the site of human cleavage in the fallopian tube only. Just after the fertilization, the fertilized egg started moving downward in the fallopian tube itself. It will start its division only. So the site is there in the fallopian when it is moving downward. The type, what are the types? Holoblastic, now we have seen. Unequal, we have seen that. Indeterminate and radial cleavage. Radial cleavage means what? This is based on the plane of the division. So that is what a radial plane of the division. We are following the radial plane of the division. The first cleavage in the human will start after the 30 hours. Now my dear child, whenever the 30 hours, 60 hours, 72 hours, 3 days, 4 days, 5 days, 10 days, whatever the duration is being mentioned, so that duration is in reference with the time of fertilization. When fertilization occurs, that time the process of embryonic development begins. So whatever the duration, suppose we are taking the 40 weeks of the gestation period. So 40 week means what? From the day of fertilization. So first cleavage in the human will start after the 30 hours of the fertilization. That is from the meridional axis. What is meridional axis? Meridional axis means what? The vertical axis which runs from the vegetal pole to the animal pole. So cleavage, the first cleavage in the human, that will start after 30 hours in the fallopian tube only. That is meridional. Remember, this is very important question, and when the higher question or the higher order questions are there, then only such kind of the questions will be there. So first cleavage in the human is runs after the 30 hours, and which is meridional in nature. Meridional in nature means to say when the cleavage plane or cleavage furrow runs from runs in between vegetal and animal pole directly or vertically. It is running in between the animal pole and the vegetal pole. So is it clear? The second cleavage just after the second duplication of the time, that is 60 hours. Second cleavage will start or complete at the 60 hour, and that is also meridional, the previous one. Third cleavage is at 72 hours. Now the duration is reduced, and that is horizontal. That is horizontal from the previous one. First one at 30 hours, second one will complete at 60 hours. Both of them are meridional. But when these two are meridional but they are opposite to each other, means what? If one plane is running from the left side, so another one will work from the right side. The third cleavage plane is third cleavage is completed at the 72 hours, and that cleavage occurs horizontal, or we can say the perpendicular to the previous one, or we can say the right angle to the previous one. And due to these three cleavage, an 8-celled embryonic structure is being produced. First 2, second 4, and third one is 8. So first cleavage will result in 2 blastomere, second will result in the 4 blastomere, and third one will result in the 8 blastomere. So 8-celled stage of the embryo is being produced. And here one very important point is being observed. At this stage, the embryo is completely a solid structure. Solid structure means all the cells are being packed tightly, all the blastomeres are packed tightly for the purpose of the cohesion. So all these blastomeres are packed tightly with the help of the protein deposition, and the arrangement of such arrangement of the blastomeres where they are arranged in form of the compact arrangement, that is called as compaction. So here the blastomeres are packed with the help of protein deposition. This is called a compaction. What is compaction? The compact arrangement of the blastomere with the help of proteins deposited in between the blastomere. So is it clear? Now moving on to the next topic and the final of the cleavage. So cleavage का importance क्या होता है? It restores the cell size and the nucleocytoplasmic index. सेल साइज मेंटेन करना, न्यूक्लियोसाइटोप्लाज्मिक इंडेक्स को रीस्टोर करना, दैट इज द इंपॉर्टेंस और सिग्निफिकेंस ऑफ द क्लीवेज. It is going to produce the multicellular embryonic structure from a unicellular zygote. Are you getting my point? Just now we have discussed 8-celled embryo is being produced by the division, continuous division, and division will start and will continue till the formation of the embryo only. So here restoring the cell size and the nucleocytoplasmic index, and multicellular embryo will be produced from a single celled zygote. So that is what the importance of cleavage. So students, after completion of the cleavage, now the embryo is going to convert in form of a solid ball like structure. That solid ball like structure is called as morula, and the formation of morula is called as morulation. So are you getting my point? After completion of the cleavage, the embryo is going to convert in form of the solid ball like structure. Just now we have seen the process of compaction. Due to the compaction, the protein is being deposited in between the blastomere, and the embryo appears as a solid structure. That solid structure is called as morula, but that 8-celled structure is not undergone or is not considered as morula. One more division is required. After one more division, that 8-celled embryo is going to convert in form of the 16-celled embryo. So cleavage result in a solid ball like embryo, that is called as morula. What is morula? The cleavage result in a solid ball like embryo, that is called as morula. That is 16-celled embryo. Means 16-celled embryo means to say, till now we have seen the 8-celled embryonic stage and they are being compacted. The process is called as compaction, and due to one more cleavage in that compact 8-celled embryo, the 16-celled structure is produced, that is called as morula. So morula word is originated from a Greek word morum. Morum means to say the mulberry. Morum is verbal meaning the mulberry. So that morula stage seems like or appears like or looks like a mulberry fruit. So that is why this is called as what? The morula. So this is formed, or the morula stage is completed during the 4 to 6 days. 4 to 6 days means what? After the fertilization. Again I am repeating, whatever the duration will be discussed now, that duration will be in term of the fertilization, and that is also formed in the fallopian tube only. The important point, remember, the corona radiata get digested during this phase. Till now whatever is present, what is present there? Corona radiata, zona pellucida, and egg inside that. So corona radiata is digested, only zona pellucida is present after the morula stage. Zona pellucida is present, remember, corona radiata is digested here and zona pellucida is present here. So just see here, what is morula? So all the blastomeres are tightly packed here, and these blastomeres are tightly packed with the process of compaction. That solid ball like structure is called as morula. Moving on to the next structure, next embryonic form, that is called as blastula, and the formation of blastula is called as blastulation. Blastula and blastulation. The formation of blastula is called as blastulation. Normally in all the animals, the morula is going to convert in a hollow cavity structure, in a hollow wall like structure, that is called as blastula. But in case of humans and in the mammals, that blastula is called as blastocyst. So what is blastula? Blastula is hollow ball like embryo. The morula was solid ball like embryo. So but what happens in the next phase, the morula is going to develop a cavity in between it. That cavity is called as blastocele. So when the cavity is appear inside the embryo, that time that is called as blastula. But in case of humans and the mammal, that is not used as the blastula, that is being termed as the blastocyst. So formation of the blastula or blastocyst is called as the blastulation. The mammalian blastula is called as blastocyst. And how the blastula is being identified? So blastula is identified by a cavity that just now I discussed. The morula is the solid ball like structure, while the blastula or blastocyst is a hollow ball like structure. When a cavity present inside, हम एक टाइट बॉल की बात करें, जैसे आप रबर बॉल की बात करते हो, एक स्पंज बॉल की बात करते हो, so rubber ball or the cork ball is the morula. When we are just discussing about a plastic ball or tennis ball, so only peripheral layers are present and the internal layer is completely vacant. So that is what the blastula or blastocyst. So is it clear? अंदर कैविटी खाली है तो that is called as blastula, और बाहर की तरफ कैविटी, बाहर की अगर कैविटी नहीं है, absent है वहाँ पे, the complete mass is arranged with the blastomere only. So that is called as what? Morula. Moving on to the types of the blastula. First type of blastula is called as coeloblastula. Coelocele means cavity, blastula. So when the blastocele cavity is present inside the blastula, then it is called as coeloblastula. Cele cavity, blastula. So when the blastula is having the blastocele present, example of the coeloblastula is the frog. Remember that point I am just discussing for the NEET examination. Coeloblastula, cele means the cavity, blastula means the blastula. So when blastocele cavity is present inside the blastula, then it is called as coeloblastula. Moving on to the next type. In some of the animal, the blastocele cavity is not able to develop, or blastocele cavity is not developed inside the blastula. So that type of blastula is called as stereoblastula. So solid blastula is called as stereoblastula. Example of that is Coelenterata, Annelida and Mollusca. Stereoblastula, the solid blastula. Common example is Coelenterata, Annelida and Mollusca. Next one is the superficial blastula. Superficial blastula, you can understand the superficial cleavage. So from the superficial cleavage, when the centroacetal egg is there, so only the top level of the egg is going to be converted in form of the blastula. So that is present in the insect only. Discoblastula, when the discoidal cleavage was there, so produced by the discoidal cleavage, prototherian reptiles and the birds are the example of that. So coeloblastula in the frog, stereoblastula in the coelenterate, annelida and the mollusca, superficial blastula in the insect produced by the superficial cleavage, discoblastula produced by the discoidal cleavage and present in the prototherian reptiles and the birds. Now we are moving to understand how the development of the blastula stage occurs, how the blastulation process occurs, how blastula is being developed. So just starting with the mechanism of the blastulation, or how blastulation is there. So mechanism of blastulation we are just going to understand. So try to understand the mechanism, just listen carefully. What happens as morula moves downward into the uterine cavity, it just going to touch to the endometrial linings, and endometrial linings are highly nutritious and they are vascular enough so that they can provide the nutrition for the developing embryo. So the morula, when it touches the surface of uterine endometrium, so here you must understand the process of blastulation is being completed there in uterus, not in the fallopian tube. So the journey of fertilized egg is completed, is reached in the uterus only. So here in uterus, from endometrial lining, the peripheral layer of the blastula cell, peripheral layer of the morula cells are going to absorb the nutrition. Absorption of the nutritive fluid from the peripheral layer of the morula cells from the endometrium. So morula cells, peripheral layer की सेल या morula की peripheral cell जो सबसे बाहर की सेल होती हैं, वो यूटेराइन एंडोमेट्रियम से न्यूट्रिशन लेना शुरू कर देती हैं. जैसे ये न्यूट्रिशन लेना शुरू करती हैं, दिस peripheral cells of the morula get enlarged enlarges and flattens up, बड़ी हो जाती हैं, चपटी हो जाती हैं, and these enlarged and flattened up cells are called as trophoblast or trophoectoderm. So are you getting my point? The peripheral cells of the morula are going to enlarge and flatten up by absorption of the nutritive fluid and will form a new type of cell. That cell is called as trophoblast and trophoectoderm. My dear child, remember this cell is very important. Tropho word once again originated from trophic nutrition. Blast means blastula. So the structure which is going to provide nutrition for the blastomere, that is called as trophoblast. Trophoectoderm, why ectoderm? Trophic layer are the cells which are going to present on the peripheral layer, that is why they are called as the trophoectoderm. And remember, these cells are not going to participate directly, or they are not going to involve directly in the process of embryonic development, but the embryonic development cannot be completed without these cells. These cells are very much important in assisting the embryonic development. They are not going to develop the embryo. Remember, trophoblast and trophoectoderm are the cells which are not going to develop the embryo directly, but then too they are important since they are going to assist in the process of embryonic development. इनके बिना embryonic development नहीं हो सकता है, but ये embryo का development भी नहीं करती हैं. So here what happens? The trophoblast is being produced. Trophoblast, the peripheral layer of the morula cells, absorbing the nutrient, and they are just enlarging and flattens up. That's why they are called as trophoblast or trophoectoderm. These trophoblasts are continuously driving the nutritive fluid from the peripheral lining of the endometrium. That is means what, from endometrium they are driving the nutrition, और उस fluid को लगातार डालती जाती inside the blastula, inside the morula, and due to that accumulation of the fluid, now what happens? A fluid filled cavity is being produced inside the morula. Are you getting my point? These trophoblast cells are continuously absorbing the nutritive fluid from the endometrium and going to release out, or going to create a cavity inside the morula. That cavity is called as blastocele. And my dear child, when blastocele appears, the embryonic stage is now converted in form of a hollow ball like structure, that is called as blastula. क्या हुआ? ट्रोफोब्लास्ट ने लगातार फ्लूइड को एब्जॉर्ब किया और उस फ्लूइड को डाला अंदर की तरफ इन द मोरिला, और उसके कारण एक कैविटी डेवलप हुई. दैट कैविटी इज कॉल्ड एज ब्लास्टोसील. और जैसे ही ब्लास्टोसील बना, नाउ द एम्ब्रियोनिक कंडीशन और एम्ब्रियोनिक स्ट्रक्चर इज कॉल्ड एज blastula. We are just going to understand some more cells of the blastula here, or blastocyst here. Remember, अभी हमने देखा था बाहर के cells, the outermost layer of the morula is going to form the trophoblast layer. All the cells which are present just below the trophoblast are going to form the inner layer of the embryo. Are you getting my point? Peripheral layer that is not involved in the that is not directly involved in the process of embryonic development, but the inner layer of the cells are having responsibility to develop the complete embryo. That's why they are called as embryoblast. Are you getting my point? The cells which lying just below the cells which are lying closer to, or just the cells lying below the trophoblast are having responsibility to develop the embryo, having responsibility to form the embryo, and that's why they are called as embryoblast, not the trophoblast. Trophoblast is providing trophic nutrition, and embryoblast, the blastomere which produce the embryo. So embryoblast, and these embryoblasts are lying just below the, or below to the trophoderm or trophoectoderm or trophoblast. That is why they are called as ICM, inner cell mass. The blastomeres present below the trophoblast, blastomere present below the trophoblast are called as inner cell mass. Inner, नीचे, ट्रोफोब्लास्ट के नीचे मिलने वाली सेल्स के ग्रुप को हम क्या बोलेंगे? ट्रोफोब्लास्ट के नीचे मिलने वाले ब्लास्टोमेयर के ग्रुप को हम बोलते हैं इनर सेल मास, और क्योंकि इनके थ्रू फॉर्मेशन होता है एम्ब्रियो का, दैट इज व्हाई दे आर कॉल्ड एज एम्ब्रियोब्लास्ट. एक और पॉइंट है एम्ब्रियोनल नाव. एम्ब्रियोनल नाव का मतलब ये होता है कि जैसे ब्लास्टोसील का डेवलपमेंट हुआ, तो ऐसा नहीं होता द कंप्लीट एम्ब्रियो और द कंप्लीट एम्ब्रियो इज filled with these cells, filled with the embryoblast or filled with the ICM inner cell mass. कुछ एक एरिया ही रह जाता है एम्ब्रियो का जहां पे ये इनर सेल मास चिपका हुआ होता है ट्रोफोब्लास्ट के साथ में. आर यू गेटिंग माय पॉइंट? जैसे ब्लास्टोसील का फॉर्मेशन होगा, तो दिस इज वेरी मच अंडरस्टूड पॉइंट कि ऑल द ब्लास्टोमियर्स और ऑल द एम्ब्रियोब्लास्ट कैन नॉट बी फाउंड टू बी अटैच्ड टू द ट्रोफोब्लास्ट और ट्रोफोएक्टोडर्म केवल कुछ पॉइंट पे ही ये ट्रोफोएक्टोडर्म से अटैच होके रहेंगे. सो जिस पॉइंट पे दे आर फाउंड टू बी अटैच्ड और लाइंग क्लोजर टू द trophoblast, that point is going to form a projection that is called as embryonal knob. So ICM comes to lie as the एम्ब्रियोनल नॉब ऑन वन साइड. एक तरफ जाके वो एम्ब्रियोनल नॉब के फॉर्म में अरेंज हो जाता है. आईसीएम एक तरफ एम्ब्रियो के जाके एम्ब्रियोनल नॉब के फॉर्म में अरेंज हो जाता है. एक और इंपॉर्टेंट क्वेश्चन आता है, सेल्स ऑफ रॉबर्स पूछा जाता है. सो ट्रोफोब्लास्ट सेल्स विच आर इन कॉन्टैक्ट ऑफ द एम्ब्रियोनल नॉब दे आर कॉल्ड एज द सेल्स ऑफ रॉबर्स. सेल्स ऑफ रॉबर्स मतलब अभी अपन डायग्राम से भी समझ लेंगे. सेल्स ऑफ रॉबर्स दैट आर लाइंग क्लोजर टू द trophoblast cells that are lying closer to the embryonal knob, that is called as the cells of Robert. Just try to understand with the help of this diagram. Here what happens, the embryonic structure you can see, the complete blastocyst is being shown. The peripheral layer of the cell is called as trophoblast or trophoectoderm, and here due to the trophoblast or trophoectoderm, here inside a cavity is present that is called as blastocele, and the inner cell is called as inner cell mass. But due to the blastocele we cannot say that cells will be present there complete in the complete embryo. तो ये क्या करती है, एक तरफ शिफ्ट हो जाती हैं. सो ये एम्ब्रियोब्लास्ट जो अंदर की तरफ शिफ्ट हो जाती हैं वो क्या बनाती है, एम्ब्रियोनल नॉब का फॉर्मेशन करती है. ये पूरा का पूरा ग्रुप ऑफ सेल क्या कहलाएगा, इनर सेल मास. द कंप्लीट ग्रुप ऑफ सेल इज इनर सेल मास and that is going to form the embryonal knob. And here some of the cells of embryonal knob are going to come in the contact with the trophoblast. बाकी जगह पे ये trophoblast से कांटेक्ट में नहीं है, केवल एक ही जगह होता है जहाँ पे trophoblast के कांटेक्ट में रहती हैं. that portion or that cells which are come in the contact of the embryonal knob, the trophoblast cell which are in the close contact of the embryonal knob, they are called as the cells of Roberts. Is it clear? cells of Roberts means what? ये स्ट्रक्चर क्लियर हो गई. बाहर का स्ट्रक्चर ट्रोफोब्लास्ट हो गया, अंदर ब्लास्टोसील हो गई. ब्लास्टोसील के कारण जो इनर सेल मास था वो एक तरफ खिसक गया, एम्ब्रियोनल नॉब का फॉर्मेशन करेगा. और ट्रोफोब्लास्ट एम्ब्रियोनल नॉब के आसपास जो भी चिपकी हुई होंगी उनको अपन क्या बोलेंगे, सेल ऑफ रॉबर. एक लास्ट पॉइंट अपन देखते हैं. ट्रोफोब्लास्ट में फिर से एक पेरिक्लाइनल डिवीजन होता है. पेरिक्लाइनल डिवीजन आप जानते हैं, पैरेलल टू द प्रीवियस डिवीजन. उस पेरिक्लाइनल डिवीजन से सेल्स के दो लेयर बन जाते हैं. ड्यू टू द पेरिक्लाइनल डिवीजन इन द ट्रोफोब्लास्ट सेल, टू layers आर being produced फ्रॉम दैट ट्रोफोब्लास्ट लेयर. फर्स्ट लेयर which is present outside, that is called as syncytiotrophoblasts. Syncytiotrophoblasts. The peripheral layer is called as syncytiotrophoblasts. That is outer and it is responsible for releasing the SCG. SCG का रोल आपने देख लिया है. The role of SCG is to produce the responsible for maintenance of the pregnancy. That is very much important in the pregnancy and that has been used for detection of the pregnancy. In the previous lecture only we have seen. So trophoblast का periclinal division होगा, दो parallel seat of the cell बन जाएंगी. बाहर की seat of cell को अपन क्या बोलते हैं, syncytiotrophoblasts. That is going to release out the SCG और ये SCG प्रेगनेंसी को मेंटेन करने का काम करता है. दूसरा लेयर होता है साइटोट्रोफोब्लास्ट, जो नीचे की तरफ रहेगा, इनर वन. द सेकेंड सीट ऑफ द सेल ऑफ द ट्रोफोब्लास्ट इज कॉल्ड एज साइटोट्रोफोब्लास्ट, दैट इज इनर वन. सिंसाइटोट्रोफोब्लास्ट इज द अपर वन, अपर सीट ऑफ द ट्रोफोब्लास्ट. The cytotrophoblast is the lower seat of the trophoblast, that is inner one. And remember, these two structures are very important. Once again I am repeating, trophoblast is not the structure which is going to develop in form of embryo, but embryo cannot be developed without the trophoblast. The above two layers means what, syncytiotrophoblasts and the cytotrophoblast are going to form the chorion, amnion and the fetal part of the placenta. These two layers, syncytiotrophoblasts and cytotrophoblast, are going to form the amnion and chorion. My dear child, will discuss these layers or these structures. Amnion and chorion are considered as the extra embryonic membrane. They are assisting the development of embryo. And the fetal part of placenta also. Placenta is a connection between the developing fetus and the mother, that is going to use for the exchange of the material from mother and the fetal body. So this is what the connection between, or this is what the placenta. So fetal part is being constituted by these two layers only. Trophoblast is going to constitute the fetal part of the placenta. So I think it is clear to you all. So once again I am repeating the process of or mechanism of the formation of the blastocyst or blastula. What happens, the Morula cells or the peripheral layer of the morula is going to absorb the nutritive fluid from the endometrial lining, और उस nutritive fluid के absorb करने के कारण peripheral layer of the मोरिला सेल्स आर गेटिंग इनलार्ज एंड फ्लैटन, और उन सेल्स को अपन क्या कहते हैं, ट्रोफोब्लास्ट या ट्रोफोएक्टोडर्म। और ये ट्रोफोएक्टोडर्म अब लगातार फ्लूइड को एब्जॉर्ब करता है फ्रॉम द एंडोमेट्रियम एंड सीक्रेट्स इनसाइड द कैविटी ऑफ द इनसाइड द मोरुला, एंड ड्यू टू दैट अ कैविटी इज बीइंग प्रोड्यूस्ड दैट इज कॉल्ड एज ब्लास्टोसील. दैट ब्लास्टोसील इज when the blastocele appears, that time the morula is converted in form of the blastula. कुछ सेल्स के नाम हमने देखे थे. वन इज कॉल्ड एज द इनर सेल मास आर द एम्ब्रियोब्लास्ट. ट्रोफोब्लास्ट ऊपर की सेल है और उसके नीचे की जितनी भी सेल्स होंगी, ब्लास्टोमियर उसके नीचे के जितने भी होंगे, ऑल द ब्लास्टोमियर्स लाइंग बिलो द ट्रोफोब्लास्ट, पेरिफेरल लेयर के नीचे जो भी मिलेंगे वो सब के सब कहलाएंगे एम्ब्रियोब्लास्ट, बिकॉज दे आर हैविंग रिस्पॉन्सिबिलिटी टू डेवलप द एम्ब्रियो. और क्योंकि ये नीचे की तरफ है टू द trophoblast, that is why they are called as inner cell mass. Embryonal nerve, due to the appearance of the blastocele cavity, the inner cell mass lying to attach on one direction only to the trophoblast, so that portion is appears in form of a knob-like structure, that is called as that is called as the embryonal knob. Cell of Robber, the trophoblast which is lying closer to the embryonal knob. Then we have seen the structure. Trophoblast is going to divide in two forms, syncytiotrophoblast and cytotrophoblast. Syncytio and cytotrophoblast will form the chorion and amnion and the fetal part of the placenta, and here in the diagram only you can understand well the trophoblast, blastocele cavity, inner cell mass, the cell of Robber. So this is what the blastula stage or blastulation. So we have completed till now three major processes: the cleavage, post fertilization event में embryonic development we are observing. So first of all we have observed the cleavage. After cleavage what happens? The repeated mitotic division, multicellular embryo is being formed, that is 8 cell structure. वो 8 cell structure में one more cleavage is taken place. That cleavage is going to result in form of an 16 cell solid mass, that is called as morula, and formation of morula is called as morulation. And after morulation the morula is going to convert in form of blastula or blastocyst, where the blastocele cavity is being present. That cavity is important because that cavity is having the nutritive fluid inside for the embryonic development. So this is what we have discussed today. From tomorrow, from next lecture we will discuss the next phases of embryonic development. Now the blastocyst is ready for getting attached to the uterine endometrium. So in the next lecture we'll discuss how this blastocyst is going to attach to the uterine endometrium. Till the time, thank you.